Hi, my name is Colin Dempsey. I'm currently doing my PhD in neuroscience at Trinity College Dublin. My project focuses on the effects of environmental enrichment on neuroinflammation. Now this sounds a little confusing, so I'm just going to explain.
Inflammation occurs when the body is damaged. For example, when you cut yourself. The area around the cut becomes red and hot, and cells in that area release chemicals which prevent infection. A very similar process occurs in the brain when something dangerous or harmful is detected. Cells in the brain called microglia detect threats and release chemicals to neutralize them. Sometimes these cells can overreact and can damage the delicate neurons of the brain. This occurs in many harmful neurological disorders such as Alzheimer's disease and Parkinson's disease.
What does environmental enrichment have to do with this? Well, first let me explain what environmental enrichment is, with the help of my ratty friends.
Environmental enrichment is the process of housing animals in enriched environments. These are rats. Here they are being held in standard lab conditions. Notice that they don't really have anything to engage with or play with. In these conditions, their brains are not being stimulated. However, if we move them into an enriched environment, then their brains will receive plenty of stimulation. This is clearly much better for the animals. Environmental enrichment can be done in a number of different ways, for example the inclusion of toys, mazes, enhancing cage size, or adding extra levels. This is regularly changed to maintain novelty. Environmental enrichment, despite seeming like a simple process, has a number of profound effects on the brain. Environmental enrichment can improve learning and memory, enhance the birth of new neurons, and help protect the delicate neurons against cell death. Environmental enrichment has even been shown to prevent the onset of damaging diseases, such as Parkinson's disease. A number of different mechanisms have been shown to underlie environmental enrichment. These include induction of the neurotrophic system, a system which produces chemicals which directly help and protect the neurons, and also the increase in new neurons, which I mentioned earlier. However, although much work has been done on these mechanisms, very little research has looked at the effect of environmental enrichment on neuroinflammation. Potentially, environmental enrichment could be reducing harmful neuroinflammation, which is common in the aged population, and this in turn could be contributing to the beneficial effects observed. For my project, we compared 3 different groups of rats: a young control group held in standard conditions, an aged group held in standard conditions, and an aged group held in environmentally enriched conditions. After a specific duration, we began analysing the levels of neuroinflammatory markers present in the brains of these rats using a number of different biological techniques. Due to advances in modern medicine, people are living far longer than previously. This brings with it a whole host of new problems, including a large increase in the number of people developing neurological disorders. Therefore, research and therapies which may slow down the progress or prevent the initial development of these diseases is of the utmost importance.
If we can better understand the underlying mechanisms of environmental enrichment, we can hopefully develop better therapies and co-therapies for use in the field.